Namaste viewers. Today I'll be discussing with you isolation of the genomic DNA from animal tissue. Source will be goat liver sample. Before I begin today's class, I request you to subscribe my channel to support. Okay, I have written here experiment number 2 because in the previous video I explained about isolation of genomic DNA from plant tissue, right? So now second experiment, that is isolation of the genomic DNA from the animal tissue. Okay, in the exam question will be asked like this: isolate the genomic DNA from animal tissue. Okay, here we'll be using the goat liver sample as the source. What is the aim of the experiment? To isolate the genomic DNA from animal tissue. Okay, now let us go through the principle behind this experiment. The isolation of DNA usually begins with the lysis of cells or tissues in order to destroy the protein structures and allows the release of nucleic acids from the nucleus. Okay, the tissue for the extraction of the DNA is suspended in saline sodium citrate solution, in short SSC, and homogenized. On centrifugation, the supernatant is rehomogenized with SSC solution. The sodium citrate helps in maintaining the salinity and the SSC helps to maintain the salinity since the pH of the SSC solution is 7.4. At this pH, the nucleoproteins are insoluble while all the proteins remain insoluble. So all the proteins remain soluble, and that will be easy for the separation. Okay. The presence of the sodium citrate inhibits the activity of the enzyme deoxyribonuclease, which would otherwise degrade the DNA. Okay, as the sediment is suspended in 2 molar NaCl solution, it releases the DNA from complex and keeps it in solution. After centrifugation at maximum speed for 10 minutes, then fibrous fight DNA precipitate out on addition of chilled ethanol. Okay, this is just a basic information about this isolation of genomic DNA principle. There are so many other methods are available for this isolation of genomic DNA, but this by using this SSC also, this is one of the method will be following at the academic level. Okay, other methodology also I'll explain in my another video. Now let us go to the reagents required. We need cooling centrifuge, 2 molar NaCl solution, 0.14 molar NaCl solution, saline sodium citrate solution. Then saline sodium citrate solution preparation. This is prepared by mixing equal volumes of 0.14 molar NaCl and 0.02 molar sodium citrate solution. Then tissue homogenizer we need, then chilled ethanol. So okay, these are some of the reagents required to perform this experiment. In my previous video, I've already explained in isolation of genomic DNA from plant tissue. Okay, in detail I've explained even the chemicals why we are using in the genomic DNA. Okay, you can go through that also. Now here, ethanol precipitation usually by ice cold ethanol or isopropanol, whichever is available in your department, you can use that one. Okay, since DNA is insoluble in these alcohols, it will aggregate together, giving a pellet upon centrifugation. Okay, the precipitation of DNA is improved by increasing of ionic strength, usually by adding sodium acetate. Okay, then salt we can also use in laboratory like NaCl and KCl, whereas here the cations, that is sodium ions or potassium ions, binds to negative phosphate groups of DNA and makes it more stable in aqueous solution. Okay, in the absence of Na plus and K plus, that is potassium, DNA molecules repel each other and do not allow grouping of DNA molecules. Okay. Okay, here you can see the cell lysis to get cell extract lysate. Okay. Then once the cells are lysed, you will be able to take out the inner content of the DNA from the cell debris. Okay. Now let us go through the procedure. Very basic, simple procedure. You can follow this one. You will get very good amount of DNA. Okay. Now what you will do first you need to weigh about 2 grams of liver tissue and crush it with the help of mortar and pestle. You need to take the chilled mortar and pestle to keep that in the refrigerator. Take out the mortar and pestle and transfer this 2 grams of liver tissue, grind nicely, homogenize nicely, okay? And suspend the tissue in 8 ml of SSC solution and homogenize it with the help of a homogenizer, okay? After homogenization, you need to transfer the homogenized content to the beaker by using the a muslin cloth. Okay, and to take you take the when to use only the filtrate. So take the beaker. On top of that, you need to cover to take the muslin cloth, and you need to filter. Okay, once you filter that one, that filtrate, you need to subject for the centrifugation. Okay, then centrifugation at 3000 rpm for 10 minutes. After centrifugation, you'll be able to see the billet and the supernatant upper region, right? Discard the supernatant and suspend the billet in 4 ml of 2 molar sodium chloride solution. Okay. After that, again, to that billet, we're subjecting for the centrifugation with the 2 molar sodium chloride. Centrifugation at 10,000 rpm for 10 minutes. Next, discard the pellet and retain the supernatant. Okay, then measure the volume of the supernatant obtained. Okay, and to the supernatant add twice the volume of chilled ethanol, and that time you'll be able to see the presence of fine thread DNA can be seen in that particular glass tube. You need to, if you want to observe, means you can't use that plastic centrifuge tube. Okay, you need to transfer that supernatant to the glass test tubes. Then to that glass test tube, whatever amount of supernatant you got, no, to that twice the volume of that supernatant, you need to add the chilled ethanol. Okay, and lightly if you shake, you'll be able to see the fine thread of the DNA molecule. Then you can write the result. The fibrous white precipitate of DNA is observed on addition of ethanol to the supernatant. Result is very important. Repeatedly I'll be telling, but still many students, they'll miss writing the result. When you miss writing the result, you will lose 1 mark. So without fail, you need to write result. You need to write the result. So this is all about the isolation of genomic DNA from the animal tissue. Okay, when you observe, no, this is in the test tube, you'll be able to see the DNA sample like this within the particular chilled ethanol with the supernatant. Hope today's video will be useful to you. I, Dr. Prathibha K, signing off for today. Stay tuned with me, stay safe, stay connected, and thank you for watching. Before I turn off, kindly go through this one. When you focus problem, you lose the good. When you focus on goal, you lose your problem. Think about this. Thank you, everyone.